Hello friends, in this video we are going to discuss about horizontal gene transfer in bacteria. First of all, what is gene transfer? Gene transfer is movement of genetic information between organisms. There are two types of gene transfer. Horizontal gene transfer or HGT and second is vertical gene transfer, that is VGT. During the horizontal gene transfer, gene is transferred between two independent organisms. It takes place between same or different species. Mostly, it occurs between bacterial species, but it is rare in humans and other multicellular organisms. It is also known as lateral gene transfer. Second is vertical gene transfer. In this, gene is transferred from parents to offsprings. Example, sexual reproduction in eukaryotes. Binary fission that occurs in prokaryotes is also example of vertical gene transfer. Here we will discuss about horizontal gene transfer in bacteria. First of all, advantage or importance of horizontal gene transfer in bacteria. By horizontal gene transfer, bacteria acquire large DNA sequences from another bacterium. Hence, recipient bacteria are able to respond and adapt to new environment rapidly. So what we can say, it is an important factor in bacterial evolution. On the other hand, mutation occurs relatively slowly. Also, most of the mutations are harmful. Horizontal gene transfer, on the other hand, causes large-scale changes in bacterial genome. Example, 20% of genome of E. coli is originated from horizontal gene transfer. But this horizontal gene transfer has certain disadvantages. It is the primary mechanism for the spread of antibiotic resistance in bacteria. Antibiotic resistance genes are transferred from donor to recipient. Spread of antibiotic resistance genes is becoming a challenge in medical field. Because if any infection is caused by antibiotic resistance bacteria, then it is difficult to treat that infection with particular antibiotic. Now, the general features of horizontal gene transfer. There is no direct cell-to-cell contact. Transfer DNA is naked or naked DNA is transferred in horizontal gene transfer. Horizontal gene transfer is bidirectional. There is no polarity in gene transfer because any of the bacteria act as a donor or recipient and vice versa. Transferred DNA is sensitive to DNA's activity. DNA is an enzyme which degrade naked DNA. So what we have seen, genetic material is transferred from donor to recipient. How? The recipient bacteria is able to take the genetic material. It is due to the competence of the cell. What is competence? It is ability of recipient bacterium to take up the naked DNA. And the cell which shows competence is known as competent cell. DNA that enters the recipient cell is called exogenote. Now let us discuss the generalized mechanism of horizontal gene transfer. Endogenote from donor is replicated and excised. Then it enters the recipient cell, mostly of the same species and sometimes of different species. Initially, genome of recipient is known as merozygote, partial diploid or merodiploid. So when the endogenote enters the recipient cell, initially it is known as merozygote, partial diploid or merodiploid. This endogenote of the donor is known as exogenote of recipient because it is coming from outside. Then this exogenote of recipient cell has two possible fates. First, it is either incorporated into the host genome or it is degraded by host enzyme, which is DNase enzyme. Incorporation of the exogenote into host genome occurs through two methods, recombination or insertion. So what is recombination? It is regrouping of genes, that is donor alleles, are substituted for recipient alleles. And it occurs when donor DNA segment and recipient DNA segment are homologous. This is possible when donor and recipient are of same species or closely related species. What is insertion? Donor genetic material is embedded between existing genes of the recipient genome. This happens when donor DNA segments and recipient DNA segments are non-homologous or share no homology. We will discuss these in detail in later videos. One important point that we should remember is probability of incorporation of exogenote increases if exogenote and genome of recipient share homology, or both exogenote and recipient genome are derived from same species, or in other words, donor and recipient are derived from same species. If no selective advantage is provided by transferred gene to the recipient bacteria, they are usually lost by deletion. So if the transferred gene provides no selective advantage to recipient bacteria, then they are lost. There are three methods or mechanisms of horizontal gene transfer in bacteria. First, transformation, second, conjugation, and third, transduction. These we will discuss in detail in series of videos. Hence, we can conclude that through horizontal gene transfer, new genes are spread rapidly compared to mutation, and larger, fitter genomes are achieved over a period. This is all for today's video. In the next video, we will discuss about transformation in bacteria in detail. If you like this video, please hit the like button, share it and subscribe my channel. Thank you.